Hello, it's Dr. Drredfrizzle here. What I'd like to do today is I'd like to elaborate on the idea of like the sizes of various structures within cells. So we haven't really done much on cell structure so far. So I think the ideal series to do this in will be this one, because in my last video I discussed with you how to work out sizes of particular things. So uh, I think the diagram of the cell, we could demonstrate the sizes of various organelles quite easily via the system that we learned in the last video, hopefully. So I'll draw out an animal cell. Draw out quite big now. Don't panic, animal cells are very small. They're not this big at all. They're actually very small. I'll discuss in a bit more detail with you. Plant cell is about 20 nanometers, 20 micrometers, sorry, across, in diameter, that is, right across the cell. But which is quite similar to an animal cell as well. I think an animal cell is about 20 micrometers across, sorry, which you write like this. So I'll label this 20 micrometers. Now, micro is that funny little U with a long tail. Right, so I'm going to draw for you the nucleus. Which has a region inside it called nucleolus. I'll draw this in here. Here we have the cell membrane. We have a stack of things here called the Golgi Apparatus. So that's Golgi. It's for like a snowman. And ribosomes we have. Now, the ribosome, just to be clear, is just that little tiny dot, because the ribosome is very small. Vacuoles are only in plant cells. We also have something called lysosomes, which are again very small little dots. So just draw your dot, label this lysosome. L-Y-S-O-S. Lysosome. O-M-E at the end. And that's pretty much all the organelles. Oh, and there's mitochondria as well, I just missed out. So I'll just draw you mitochondria. Right, now I'll just describe to you the sizes, okay, in a little bit more detail. So first of all, the Golgi Apparatus. It is 7 nanometers thick per one of these little stacks. So 7 nanometers. Now this gives you some sort of idea in terms of scale, because we know from our last video that 1,000 nanometers makes up 1 micrometer, which means that it's 20,000 nanometers across this cell, which means this is 7 out of 20,000. So it gives you some idea of how small in comparison to the size of the cell this Golgi actually is. I'm drawing it big so you can actually see it, because it would be tiny if we actually drew it out in the proper ratio to the size of the cell. You also need to know that your mitochondria is 3 micrometers thick, so there's 3 micrometers. So now we could do a bit of a comparison. So we know that each one of these is just over twice the height of that. Okay. A mitochondria is where the energy for the cell is produced by respiration. As I've discussed respiration before, but I never really discussed where it happens, and it happens inside these things called cells. And I've discussed cells in a little bit of detail before, but I haven't gone into much detail in terms of the actual detailed structures of cells. I've actually talked about the like cell membrane, nucleus, and all the basics, the cytoplasm and that, but I haven't really gone into much detail in terms of the detailed structures. So, ribosome, the size of that is 30 nanometers in diameter. So again, we can make a comparison here. That's 30 nanometers. This is 3 micrometers, which means that this is hugely bigger than this ribosome. The ribosome is absolutely tiny in comparison to the size of the cell, whereas the mitochondria is not quite as small as that. the mitochondria is 3 micrometers. So I've actually drawn this. I think I made a mistake. 7 nanometers, 33 micrometers. No, maybe I haven't made a mistake. That just seems very strange because, think about it, 3 micrometers. That would almost make it. If we do that like that, it's about 20 of them could fit in the height of the actual cell. And this is nanometers, which means that it's 1,000. Each one of these units is 1,000 times smaller than each one of these, which means that you need 3,000 of these to make up that sort of size. So that seems very, very strange. I did actually get this information off a website, so I might have to double check. But according to that, mitochondria are much, much bigger than Golgi. Right, so Golgi, I'll talk to you a little bit about the Golgi apparatus. What this does is it assembles proteins into vesicles. So vesicles are anything that's surrounded by a membrane of some description. So you could have what you have. I haven't drawn this in. You have something called the rough endoplasmic reticulum, so I'll just abbreviate it to RER, which you can do usually. Rough endoplasmic reticulum is the word for it. And what this is it's a series of fluid-filled sacs that make it up, and they have folds known as cisternae. So I'll just write on there. Cisternae. It's spelled C-I-S-T-E-R-N-A-E. And what this does is this has little things called ribosomes on it. So these ribosomes aren't just around anywhere in the cell like I've drawn there. They can be sorry, excuse me. They can be on the RER, rough endoplasmic reticulum. Sorry, I'm problems with my throat at the moment. So basically, what these do, these little ribosomes, is they make proteins, and these proteins are transported to the Golgi, where they have basically a membrane put around them, which enables them to be transported outside the cell or to where they're needed. So what we call this, we call them vesicles. Once the protein has a membrane around it, we call this a vesicle. And an example of a vesicle is a lysosome. A lysosome is a vesicle. Vesicles are very small things. What a lysosome vesicle is used to do is it contains a protein known as lysins, which are a particular type of digestive enzyme which basically digests anything that the cell doesn't want. So supposing the cell's made a protein that's been like mutated, which means that the genetic information has been altered in some way. It could be harmful for the cell, so the cell can just digest it via these lysosomes with the lysins. The other thing I just wanted to add about this nucleolus in here, because I haven't spoken much about that. Nucleolus. This contains ribosomes, which again make proteins, like on the rough endoplasmic reticulum, and RNA. Now RNA I'll go into a bit more detail in the previous video. I don't want to talk too much about that because it's quite a complicated thing. And these little gaps here I haven't really said much about. These are nuclear pores. Nuclear pore. P-O-R-E spelt. And what these are is they're holes to enable things to move from this nucleus organelle here and to essentially just escape from the nucleus. So you get all sorts. You get like proteins. It is actually mostly proteins because most cells are just composed of mostly proteins. You can get like other nutrients and stuff. Can't think of many examples at the moment. You can get like sugars and stuff because they're needed for respiration. Yeah, there isn't much in addition to proteins, I don't think, because the majority of there's loads of things like hormones in the body, hormones, enzymes, red blood cells. They're all mainly made out of proteins. These two, hormones and enzymes, are particular types of protein. So I'd like you to familiarise yourself with the fact that they are loads of things in our bodies that are made up of proteins. Pretty much all these organelles are in here, all these particular things, and pretty much everything within the body is made up of proteins. So that's why I struggle to find another example of things that are transported in and out of the nucleus, because it's mostly proteins. And that's pretty much what I want to talk to you about, apart from, I want to throw out a little question for you. How does a prokaryote, which basically prokaryotes are cells in a more primitive form, they're like this, but they have just a little bit of DNA in the middle. You might have seen this with like viruses and stuff. They're examples of those, just because they don't have membrane-bound organelles, which basically means they don't have things like the nucleus. They just have a bit of genetic information in the middle, and they just have a membrane. They don't actually. Well, it's not always called a membrane. I mean, in a virus, for example, it's called a protein coat, but you can also call it it's just essentially a cover for the cell. It's not, and it is usually made of protein, so I'll call it a protein cover. That's what protein means. Protein cover. Now, from this, the reason why I brought this up was because from this little primitive form of cell, how does these membranes around these particular organelles, how do they actually form? Well, to answer this, we need to have a look at a few theories that I've already done my research. And why it seems to be that the general sort of verdict for people is by a process known as endosymbiosis. Now, this sounds like a really complicated, really confusing word again, but it's really not. All it means is that all the particular things, such as this, inside the cell, that there's membrane-bound organelles that are unicellular organisms, which just have one cell, that are an organism in their own right. So like this mitochondria, where's the mitochondria? There it is. So that mitochondria, for example, might have started out as its own organelle. So I'll label this mitochondria. Sorry about this, my pen's running out. Organelle, unicellular organism. So basically, this mitochondria was originally, people think, an organism all on its own. But what they think happened is they think that the cell ingested this particular organism, like a lot slightly more developed or larger cell ingested this, and then what happened is that the cell realised, this larger cell realised that having the mitochondria in it generated energy for it, so it enabled it to carry out a lot of processes that otherwise it wouldn't have been able to do. So this is how scientists think that all these particular membrane-bound organelles actually developed into a eukaryotic cell, which is the name of this cell, and this is our cells. Our cells are eukaryotic cells, and what this means is just they're basically got membrane-bound organelles, so like the nucleus, lysosomes, ribosomes, etc. So that's generally what I wanted to talk to you about in this video. In my next video, I'd probably like to discuss a little bit more about the functions of these particular organelles, and this one I'll mainly focused on sizes and stuff. Hope this has been helpful, and see you in the next video.